Hello all. In this video we are going to talk about 3-2 systems of classification. Now just like in 3-1, hopefully this will serve mostly as a review of us leading into our lower animal section. So let's begin. As we talked about in our last lecture as a class discussion, Carolus Linnaeus's form of classification and the use of his dichotomous key. A dichotomous key is a series of yes or no questions. It's designed to distinguish between similar species. So for example, if this whatever is blank, go here. Okay? So really it could be if this animal has wings, then go to number 2. If it doesn't, go to number 3, and on down the road. Okay? So the advantage of this dichotomous key is that it's easy to use, there's no formal training required, and can be easily done in the field. The disadvantage is that there is an assumption that all species are known, and that we know for a fact that there's many species on our planet that have still not been identified. And another big disadvantage is that the dichotomous keys are solely based on physical features. We've also learned throughout recent studies and new advancements in technology, specifically in DNA barcoding, which we will talk about, we know that physical features alone is not enough to decipher who's closely related to whom. Okay? So dichotomous keys, we do have some advantages. They're easy to use. They can be done in the field, but we have to have the assumption that all species are known and based solely on physical features. Cladistics. We will spend the most time in cladistics in our unit and in 8th grade. Cladistics is a study, and it's using an animal's evolutionary heritage to classify. We have a clade, which is a leaf, as if on a family tree. So as an example, this would be a clade, and these would be organisms. And then these events would be advancements, whether mostly physiological advancements. So we'll go through several examples of these here in a second. The advantage to cladistics is that it has a better chance of being truly related to one another. It is also pretty easy to use. The disadvantage is that we are dependent on fossil data, so things that have already died and passed away, assumptions made as to the function and behavior of extinct animals. So, for example, if we're trying to trace all the way back to the dinosaurs, we have to make assumptions about what each fossil limb was used for. So we do have to make assumptions, and we are dependent on fossilized data. This is an example of a cladogram. As you can see, this is a clade. Okay? And each separation, so between a hagfish and a perch, in between here we have some form of evolutionary advantage. Okay? So in this case, we're looking at in between hagfish and jawfish, like the perch, we have the advancement of jaws. Then in between perch and a salamander, we have the advent of lungs. So then I go into amphibians. Then we have a lizard, which has claws and nails, and then we have, keep going up, then we have feathers. That's a new one, a new advantage, and that gives us birds. And then fur/mammary glands, and that gives us into our mammals. Okay, so each rung up the clade or cladogram, we have some form of advantage in between the two. So as we advance going this direction through time, from simple organisms to more advanced organisms, we have different advantages. Okay, hopefully that's pretty clear. Here is another example of a cladogram for mammalia. Okay, and we can start out and branch out into our rodents. These are our apes, and I'll just give you a few seconds to digest how complicated cladograms can get. And these are all families. Okay. The last form of classification we're going to talk about is called DNA barcoding, and it really is the next generation of classification. For animals, we look at a specific region of a mitochondrial gene called COL, or cytochrome C oxidase subunit L. So I guess that would actually be COI, not a lowercase l. So COI, cytochrome C oxidase subunit I. That's the very specific. You will not need to know the specific gene that they test in animals. That will not be on a test, but I thought it would. I would get some questions as to which gene is utilized for that screening, and that is the specific gene that is used. Let's go back a slide here. Organisms are groups based on similar sequences of DNA. So we look at genes, and then specifically this gene requires an already known section of DNA to check for. So science really knows this section of DNA, this mitochondrial gene COI. It knows it very well, and that's why one of the reasons why they use it. Also requires a specialized lab and personnel, and is very helpful in the process of archaeology. And this is an example of that DNA barcoding sequence. So depending on how similar different DNA barcodes are to each other, the more similarities we have, the closer related they are. So in review, things that you need to know on. You need to understand the three main types of classification. You need to understand how they're grouped, what is the major tool used, when it's used, the advantage and disadvantages. So for your tests, it's a good idea to specifically study this particular table. So for our Linnean, this is our dichotomous key. We have physical characteristics, so groups based on the physical characteristics, and it can be described in this or that way. The tool used for Linnean classification is the dichotomous key, and is generally used when attempting to quickly identify an unknown species. The advantage is that it's very easy, no specialized equipment or training needed, and the disadvantage is that things that look alike on the outside may not be the same or do similar things internally. Okay? The next step, or the next evolution, if you will, of classification came in with cladistics, and we look primarily or group organisms based off of external or internal structures shared by certain fossils. Okay? The tool used is a cladogram. Again, it's that leaf structure where we have time going this direction, and then over time we have these advancements and different organisms evolve. Okay? So that is a cladogram, and it determines the ancestry or lineage of an organism. So that's how it's used. Another advantage of it, or the advantage of it, is that again it's very easy, just like in the dichotomous key, and it is based on similar characteristics or behaviors. Now the disadvantage is that since we're using fossils, we're never 100% certain if you have all the specimens, and that what fossils we do have, the estimations of what each function for each structure is correct. Finally, the next evolution, the most current form of classifying organisms, is through DNA barcoding. And we have similar segments of DNA in the nucleus. That's how we group organisms. We have DNA fingerprinting as the major tool that is used. And again, we extract DNA from an organism. We run it. We send it to a lab. They run a coding test called a PCR test, and then we get comparative data on the organism that we're testing and comparing it to other similarly tested organisms. So it's generally used when you are trying to prove that two organisms are related to each other. The advantage is that the relationships between two organisms is genetically confirmed, and usually it's with high degree of accuracy, something like 99%. The disadvantage is it's long, it's expensive, and it requires experienced scientists in a lab. So it's not something you can do quickly in a field like you can with a dichotomous key. Okay? So that is your lesson on classification and the three major types of classifying systems. Again, in our course, we will primarily be using cladistics. All right? Now, your video secret number is going to be 111. So 111, that is your video secret number for this lesson. Thanks so much for watching. I look forward to seeing you guys in class. Bye now.